Welcome to Educon. This is Imam with you, and today we'll be learning about biomolecules. Biomolecules includes lipids, nucleic acids, carbohydrates, and proteins. So, in earlier chapter, we study about the cellulose, what is cell, and what is the difference between the animate and inanimate and animate things, like that. So, here we'll be learning about the chemicals which are present in all living organisms. So all living organisms are made up of chemicals. Okay. If any chemical analysis today, if you perform on plant, animal tissue, or microbial paste, we obtain a list of chemical elements like carbon, oxygen, hydrogen, and several other constituents, chemical constituents in the tissue or in animal tissue or plant tissue or anything. If the same analysis is performed on earth crust, we obtain the same compound with the difference in abundance of carbon, hydrogen, and other chemicals. So for that, what we'll do a chemical analysis. When you in higher classes you learn about this chemical analysis. Here what we'll take a living tissue like plant, or you take a liver, wet liver, okay, and you grind it with the help of trichloroacetic acid. So what you'll get a thick slurry. Okay, what you'll do, strain through the cheesecloth or fine cotton like as a filter. So you will obtain two components from that thick slurry. One is filtrate, okay, which pass through the cheesecloth, and the another one is the retentate. Here the whatever the remaining material is present on the cheesecloth, it is retentate. The filtrate is the acid soluble and it contains micromolecules, okay, whereas the retentate, which cannot pass through the cheesecloth or filter cloth, it is acid insoluble pool, and also it contains macromolecules. So again, these micromolecules and macromolecules, if you perform various separation techniques, you will get the specific molecular formula of that particular compound. What is that compound? Again, all the carbon compounds obtained from living tissue is called biomolecules. Remember this, all the carbon compounds obtained from living tissue. Okay, we are not talking about earth crust here. So all the carbon compounds obtained from living tissue is called as biomolecules. Living tissue also contain inorganic elements or compounds. Okay, and till now we talk about the about the carbon compounds which are in living tissue. Now we'll discuss about the inorganic elements or compounds which are also present in living tissue. Now, for example, here we have a image. Okay, comparison of elements present in non-living and living matter. Okay, fine. Here what is there? We have an element on this side, okay, and percentage weight of earth crust. Here this is earth crust and this is human body. What we have here, hydrogen, we have around 0.14% on earth crust, whereas in human body it is higher in percentage. Okay, next, carbon. Okay, it is in the earth crust, it is 0.03, whereas in human body it is 18.5. Okay, and oxygen in earth crust it is 46.6, whereas in human body it is 65. Nitrogen is very little in the atmosphere, and in human body it is 3.3% weight of the human body. Then we have sulfur, which is also higher in human body, Then we have this sodium. From here you observe the things. 2.8 here in the earth crust, whereas in human body it is lower levels. Okay, same for calcium also, it is less in human body. Magnesium too, it is less. And coming to silicon, that is, it is 27.7% on earth crust, whereas in humans it is negligible. Okay, so here around all these 1, 2, 3, 4, 5 hydrogen, carbon, oxygen, nitrogen, and sulfur, their percentage comparing to earth crust in human body it is more. Okay, and here on the under table, this is the general representation of the compound in a formula. List of representative or the inorganic constituents of living tissue. Components and the formula. As you all know, sodium is sodium means Na plus, potassium means K plus, and calcium it's Ca plus, and so on. And regarding how to analyze the inorganic material which is present in the living tissue. What we'll do, we'll wet a small amount of leaf or liver. Okay, then we'll let it dry till the water inside is evaporated. Okay, now what we'll do, we'll take the weight, and it is called as a dry weight. Okay, then we'll burn the dry tissue, which will leave the carbon compounds to be removed as CO2. Okay, so after burning, what will happen? Because now the dehydration happened, because we let it dry till the water is evaporated. After that, we'll take a dry weight of that, and then we burn the dry tissue, that is leaf or liver, which will leave the carbon compounds to be removed as carbon dioxide. Okay, again, remaining dry ash contains inorganic elements like calcium, magnesium, etc. Okay, so apart from biomolecules, we have an inorganic constituents in our body. How we will find out that? It is by allowing the wet tissue until till the water is evaporated, to dry it, and then we'll take a dry weight of that tissue, then burn it, and then during burning, the carbon compounds to be removed as CO2 will dissipate, and the remaining ash contains inorganic elements like calcium and magnesium, etc. Biomolecules, in the starting, I told you that carbohydrates, lipids, and proteins, nucleic, all these comes under biomolecule. Here carbohydrates, we have sugars, starches, and then cellulose. This is monomer. Okay, whereas in lipids, fats and oils, then saturated fats, then unsaturated fats, and lipids also the main constituents of membranes, cell membrane. Okay, again for proteins, we have amino acid. Proteins are made up of amino acids. Okay, amino acids are made up of peptide bond. Then we have nucleic acid. We have nucleotides, bases, and pentose sugar, ribose sugar. Then we have DNA and also RNA. It is a simple representation how to understand what are biomolecules. So it will give an idea. So what comes under which comes under what. Okay. Coming to amino acids. So amino acids are organic compounds containing amino group. Okay, this is the amino group. Okay, on the same carbon, and an acidic group as a substituents on the same carbon. Amino group and the acidic group, that is the carboxyl group. Here it is there. Okay, same carbon. Then see, it is called, that is the carbon is called alpha carbon, hence called alpha amino acid. They are substituted methanes. Okay, there are 4 substituent groups occupying the 4 valency positions. Okay, Here on each carbon we have 4 valency, okay, valency positions, okay. So each valency has 4 substituents groups occupying the all the 4 carbon, all the 4 valency position. One is we have hydrogen, we have carboxyl, you can see here hydrogen, okay, carboxyl, amino group, one variable R group, okay, here variable R group. The R group may be monomer or many monomers joined together, okay. Based on R group, there are many amino acids, okay, and in protein only 20 types of amino acids are studied. Okay, right now in proteins only 20 types of amino acids are studied, okay, and based on R group there are many amino acids. Chemical and physical properties of amino acids are essentially of the amino carboxyl R group. Okay, we need to see this sentence is particularly important because we can expect a question based on the statement. For example, which of the following statement is true? Okay, now this line is most important, like chemical and physical properties of amino acids are essentially of the amino carboxyl R group. Now again another statement we will be having chemical properties of amino acids, so physical will not be there. So we need to make sure that when we are reading the NCERT book, we need to keep this new chemical and physical properties of amino acid are essentially this amino carboxyl and R group, okay, R group, okay. This is the important line. It may come based, the question may come as a statement, so find the true statement, okay. And based on the number of amino and carboxyl group, there are acidic, basic, neutral amino acids, okay. Again based on the number of amino and carboxyl group, okay, again they are acidic, basic and neutral amino acid. Here we have glycine, alanine, and here we have NH3 plus, that is amino, we have a positive charge, and COO minus we have negative charge. Here this is the, and we can also say zwitterionic form because we have a equal number of cations and anions here, okay. And this is alanine, valine, okay, leucine, methionine, isoleucine. Okay, these are non-polar aliphatic R groups, okay. Whereas polar uncharged R groups, okay, we have serine, polar, okay, polar means uncharged, the R group, this is the R group, okay. And we have a threonine, cysteine, proline, asparagine, glutamine. So these are the examples which comes under polar uncharged R group, okay. Again we have aromatic R groups, okay. Aromatic R groups, what is there? Phenylalanine, tyrosine and tryptophan. So see this, the rest of the structure is same, okay, only the variable R group is different in all the amino acids. And we have positively charged R groups, okay. Here this group, this is the pink, the shaded one, this is the R group, so positively charged R group. Here we have the positive charge is on R group and also on one of the amino group also, remember that, okay. And we have negatively charged R group. Here what happened, we have more anions than the cations also. So around non-polar aliphatic R groups, polar uncharged R groups, and then aromatic R groups, positively charged R groups, negatively charged R groups. All this, okay, comes under the amino acids. So here what we need to keep in mind is that the 4 substituents groups occupy the 4 valency positions, and the hydrogen carboxyl amino along with the variable group R, which differs in various amino acids. Only 20 types of amino acids are studied, and chemical and physical properties of amino acids are essentially of the amino carboxyl and R group, and based on the number of amino and carboxyl group, there are acidic, basic and neutral.